Welcome to Comparative Anatomy of Vertebrates. Today, we are going to explore the alimentary canal. This is not just a simple pipe inside the body, it is a highly sophisticated, evolutionary marvel that allows animals to extract life-sustaining energy from their environment. Whether we are looking at a tiny fish or a massive elephant, understanding this system is the key to understanding how vertebrates survive and thrive. To begin our journey, imagine a long, winding conveyor belt inside a highly efficient factory. Raw materials enter at one end, get processed, and waste is dumped out the back. This is exactly what the alimentary canal is. It is essentially a tube within a tube that runs through the animal's body. Every vertebrate shares this fundamental design, consisting of the oral cavity, pharynx, esophagus, stomach, and intestines. This continuous pathway is absolutely essential because it allows the animal to safely bring the outside world inside, breaking down complex foods to extract the energy needed for survival. Now that we understand the basic layout, let us look at how it forms and functions. Embryologically, the inner lining of this tube comes from the endoderm, while the surrounding muscles develop from the splanchnic mesoderm. Together, they create a dynamic system capable of ingestion, digestion, and absorption. But perhaps the most crucial feature is compartmentalization. Muscular valves called sphincters separate different regions. This allows a highly acidic stomach to safely empty into a basic intestine without damaging the organism. Without this specialized, compartmentalized tube, complex animals simply could not sustain their high metabolic rates. This structural complexity leads us to an important question. How efficiently does an animal digest its food? Biologists measure this using a concept called mean retention time. The formula is T equals V divided by R. Conceptually, this tells us exactly how long food stays inside the gut to be processed. T is the retention time in hours. V is the total physical volume or capacity of the gut in liters. R is the rate of food intake in liters per hour. If the gut volume V increases, food stays inside longer, allowing for more thorough digestion. Conversely, if the intake rate R increases, food is pushed through faster, decreasing the time available to extract nutrients. Before we move on, let us see this formula in action with a concrete example. Consider a large herbivore, like a cow, which relies on fermenting tough plant matter. We are given a massive gut volume of 120 liters and a steady food intake rate of 2.5 liters per hour. We need to find the mean retention time to understand its digestive efficiency. We chose this formula because it directly relates physical anatomy to physiological time. By substituting our given values, we divide 120 by 2.5. The final answer is 48 hours. Physically, this means a piece of grass stays inside the cow for 2 full days, giving symbiotic bacteria enough time to break down tough plant cellulose. Having calculated how long food spends in the system, let us visualize the pathway itself. Here you are seeing a generalized schematic of the vertebrate alimentary canal. Notice the unidirectional flow indicated by the arrows. Food enters the mouth on the left, passes through the pharynx and the narrow esophagus, and enters the expanded stomach where initial chemical digestion occurs. From there, it moves into the highly coiled intestine, which is the primary site of absorption, before waste exits through the cloaca or anus on the right. This diagram beautifully illustrates how the physical volume we just discussed in our formula is distributed across different specialized regions to maximize digestive efficiency. Now that we have traced the path of food, we must ask how the body actually captures the nutrients. Have you ever tried to clean up a water spill with a smooth piece of plastic? It does not work. But a fluffy cotton towel absorbs water instantly because its millions of tiny threads create a massive surface area. The vertebrate intestine uses this exact same physical trick. Maximizing surface area is literally a matter of life and death. If nutrients pass through the gut without touching the lining, precious energy is wasted. Because a perfectly smooth intestine would have to be miles long to absorb enough nutrients, vertebrates evolved folded tissues to pack massive surface area into a small abdominal cavity. Building on this need for surface area, let us look at how different vertebrates achieve it anatomically. Cartilaginous fishes, like sharks, possess a spiral valve that forces food to corkscrew through the intestine, increasing both surface area and transit time. Higher vertebrates, like mammals, take this much further. Their intestinal lining is folded into macroscopic ridges called plicae circulares. These are covered in tiny finger-like projections called villi, and the cells themselves have even smaller microvilli. This brilliant, fractal-like folding strategy increases the total absorptive surface area by a staggering factor of up to 600 compared to a simple smooth cylinder. To truly appreciate these anatomical modifications, we must look at the physics governing them. This is Fick's law of diffusion. Conceptually, it proves why surface area is the most critical factor in biological absorption. Let us break down the symbols. J is the total rate of nutrient absorption in moles per second. D is the diffusion coefficient, a constant based on the specific nutrient. A is the total surface area in square meters. Delta C is the concentration difference driving the nutrients, and delta X is the thickness of the membrane. Physically, if the surface area A increases, the absorption rate J goes up dramatically. Conversely, if the membrane thickness, delta X, increases, the absorption process slows down. Let us apply Fick's law to see exactly how poorly a simple tube would perform. We are calculating the baseline absorption rate for a hypothetical smooth, cylindrical intestine. We are given a small surface area of 0.5 square meters, along with standard values for the diffusion coefficient, concentration gradient, and membrane thickness. We use Fick's law to establish our baseline. By plugging in our values, we see that the concentration gradient divided by the thickness gives a driving force of 10,000. Multiplying this by the diffusion coefficient and our small area yields a final answer of 0.05 moles per second. Physically, this is a very slow rate that would likely lead to starvation for a large, active vertebrate. Since we just saw that a smooth intestine is inadequate, we will now mathematically prove the immense biological value of a folded intestinal lining. In this first step of our proof, we are setting out to recalculate the absorption rate using a biologically realistic surface area. We replace the initial area with a massive 300 square meters, representing the presence of microscopic villi. Notice that the diffusion coefficient, concentration gradient, and membrane thickness remain exactly the same. This is because the cellular properties of the membrane have not changed, only the macroscopic folding of the tissue. This setup isolates the mathematical effect of surface area, preparing us to see the final impact on absorption. Continuing our proof, we now calculate the final result of this anatomical modification. By multiplying our constant driving force of 10,000 by the diffusion coefficient and our new massive surface area of 300, we arrive at the enhanced rate. The math reveals a new absorption rate of 30 moles per second. When we compare this to our baseline of 0.05, the intuition becomes clear. Simply by folding the tissue, the vertebrate intestine increases its nutrient absorption rate by a massive factor of 600. This mathematical reality perfectly explains why evolutionary pressures have consistently favored highly folded, complex alimentary canals. Without these microscopic folds, high-energy animals simply could not exist. Having proven the mathematical necessity of surface area, let us look at the actual biological structures that make it possible. In this diagram, you're seeing a cross-section of a mammalian intestine. Notice the large, circular inward folds. These are the plicae circularis. As we follow the magnification arrows to the first call-out box, you can see the villi, which are the finger-like projections covering those folds. Following the next magnification arrow, we zoom in on a single cell to reveal the microvilli. This diagram perfectly illustrates the fractal nature of the gut lining. Every level of magnification reveals more folding, which is exactly how the animal achieves that massive 600-fold increase in surface area we just calculated. To wrap up our lecture, let us review the most important ideas we have covered. First, the alimentary canal is not just a pipe, it is a highly specialized, compartmentalized tube designed to safely extract energy from the outside world. Second, we learned that the physical volume of the gut directly controls the mean retention time, allowing animals like herbivores to perfectly match their anatomy to their diet. Finally, Fick's law of diffusion showed us that massive surface area is an absolute requirement for survival, driving the evolution of complex folds and villi. You now have a solid understanding of how physical anatomy and physiological survival are beautifully connected in vertebrates.